It's 2 hours past lunch, and Fred is already starting to feel hungry. But Archie could go on for hours without even thinking about food. One of the reasons for this difference is genetic variation. Everybody has a unique combination of genes, and that goes for animals too. At the Institute of Metabolic Science in Cambridge, one of our programs of research looks at eating behavior that can lead to obesity in Labrador dogs. Being overweight is quite common for Labradors and can lead to poor health, including metabolic disease. Some dogs tend to gain weight because they seem to feel hungry all the time. This can result from genetic differences as well as from differences in diet and exercise. But why does anyone feel hungry at all? An empty stomach will send chemical signals to the brain to say that more food is needed. After eating, other signals send the message that no more is needed. However, this system can go wrong. About a quarter of UK Labradors and an even higher percentage of assistance dogs have a tiny bit of their DNA missing. This is called the genetic or DNA mutation, and as a result, a protein that's part of the hunger signaling system is made incorrectly. This means the message saying that the dog's had enough food doesn't get through, and the dog feels hungry when it shouldn't. Let's meet Spot and Lucky. Lucky has a healthy weight, but Spot is overweight. We could guess that Spot has the mutation because he's overweight, but maybe it's just that his owner isn't able to take him for walks. Or maybe Lucky has it, but his owner is super strict about what he gets to eat. Our owners would like to know if their dog has a genetic disadvantage, and our research scientist would like to understand more about genetic variation related to hunger. If we get DNA from Spot's and from Lucky's saliva, we can use a gel that acts like a sieve to separate DNA pieces that are different sizes. But the size difference on account of the missing DNA is too small to detect. To check for genetic mutations, our scientist has a really useful tool called a restriction enzyme. It cuts DNA, not at random, but only when it recognizes a specific DNA sequence. If the DNA has the mutation, there's no cutting site that the restriction enzyme can recognize. After using the restriction enzyme, we can load our samples onto a gel and apply an electrical current in one direction. The gel acts like a sieve, and smaller pieces of DNA run through it faster than bigger ones. After staining the DNA, we will be able to see different patterns of bands and then compare them to find out whether either of our dogs has the hungry gene. Unfortunately for Spot, we found out that he does have the genetic mutation, so his owner will need to take extra care to restrict his diet and provide lots of exercise.